everybody. Welcome to this week's podcast broadcast of This Week in Science. Back again with all the hosts. People have rested, are on their way to recovery from the fall crud that it comes with back to school season and people. Just people. Thank you for the crud, people. Anyway, we are so glad to be here, and we're glad that you're here with us. Hit those likes, put your thumbs on the screen, or whatever, do the thumbs up, etc. Let's get in the algorithm to share this live program, and let's make sure we get out there, like, share, notify, etc. Tell people to subscribe, get them over here because we are live. This part of the show will be edited out forever. No, this is here in all of the videos, but the podcast will be edited. And we ready to go?
Yeah, let's do it.
Yeah.
Let's do it. Tight 90. Yes. Starting the show in 3, 2. This is TWIS. This Week in Science, episode number 1029, recorded on Wednesday, September 17th, 2025. Sentient meat science. You like my sibilance? Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with ghost sharks, alcoholic chimps, and ant clones. But first...
Disclaimer, disclaimer, disclaimer. Life is such an everyday thing here on Earth. Everywhere you look, in every drop of water, under every rock, in the air, under the soil, every cranny, every nook, life has found a way over billions of years. Elsewhere in the universe, not so much. What we know of life here suggests it should be out there. Suggests that whenever the conditions are right, life must occur. And not an accident, not a fluke, but a natural progression of chemistry. An almost inevitable feature of the universe, though not necessarily a permanent one. To be a multicellular, complex, sentient form of life, that is incomprehensibly rare. You are very special. Well, and while we are here doing this being alive thing as this complex sentient creature, we need to take pause and celebrate our ability to know. Because that's the really special thing about being a sentient life form, knowing things. And that's what we're going to do here on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again talking about science. We took a week off. Hopefully we're all a little bit healthier, more hale, ready for this dynamic, exciting discussion that lies ahead. We have all sorts of stories. So much has happened. I have stories about earthquakes, fighting cancer with microbes, science on Mars, ant clones, and bad math. What do you have, Justin?
That's a great question. I haven't looked yet, but I assume I've got an ancient hominin type story. Actually, I do. Homo naledi update. Social bees, drunk chimpanzees, and that's it.
That's all? No, that's not all. Blair, what's in the animal corner?
I have koala vaccines. I have matador bugs And I have ghost sharks.
I don't know matador bugs. I am looking forward to that one. And it's very rare that people see ghost sharks. It's like ghosts generally.
Well,
right?
see, a ghost has to die with unfinished business for you to be able to see a ghost shark. So it's very rare. They're very good at finishing their business. And then also it's very uncommon for humans to be where their unfinished business is, where they are tied and where they must haunt.
Yeah.
So
It's true.
it's, there's probably some more ghost sharks than we're used to seeing. No, in reality, that's an actual animal, and we'll talk about it.
I feel like if that scenario for creating a ghost was correct, that there would be like a lot of ghost dogs and squirrels.
I didn't find my nuts. Where did I put my
and
phone?
and all the dogs are still waiting for you to come home.
And play
They're like
fetch.
the
Oh,
ghost bark
stop.
every time.
Don't give me that.
Oh.
Just waiting.
Oh,
boy.
poor little ghost
walk.
dog. Well,
Can't they see me?
definitely. I've been watching too much of the old show Supernatural, so that's where I am these days. It's so much fun. But you know what? That's not real science at all. And we're going to jump into our show. But before we do, I want to remind you that subscribing to TWIS on your favorite podcast platform is the best way to catch our show weekly in your ears. Look for This Week in Science in your favorite podcast player. Also, you can find us on YouTube, Facebook, and Twitch. We are live weekly around 8 p.m. Pacific time and on Wednesdays. And those episodes are up on our channels. Look for This Week in Science. On Twitch, we are at TWIS Science. Are we ready to do this?
Always.
Give me some science stories. Immediately, I demand it.
Okay, right now, I will jump in with a story that gets those excited headlines going. Did we just find life on Mars? No, we did not.
What?
Yeah. So research out of the SETI Institute was published recently, and researchers from SETI, the SETI Institute senior research scientist Janice Bishop and University of Massachusetts engineering professor Mario Parente, took a look at evidence from the Mars orbiter. So they were looking at orbital hyperspectral images. So the orbiter looking at these using spectrometer to look at what's going on in the atmosphere, on the ground, on the surface of Mars. And the news came out, and of course it's from SETI, and it was like about Mars, and they found chemical evidence of chemical processes that are found here on Earth in relation to biological microbes. So metabolic activity, biological origins. We've been able to do the experiments here on Earth that say, yeah, these compounds come from life, from living organisms, but on Mars, we can't do that. We need to do the experiments still. But what they were able to show in a Nature article, they were able in this Nature News and Views article to explore Perseverance's confirmation of minerals being observed from orbit, and also minerals that usually aren't detectable from orbit, and these on the ground measurements and readings that we've been able to examine smectite clays and carbonates using a tool called compact imaging spectrometer for Mars. So chemicals, molecules, they have come from processes that are chemical in nature, whether that chemistry took place in a cell or as the result of cellular activity is yet to be determined. So if you've seen headlines and speculation about life on Mars,
But
I
Kiki,
don't know. No.
that's not the big life story that everybody's talking about.
No? Is there another?
Yeah, I thought that's what you were talking to talk about. So they found spotted rocks.
Yeah, they're rocks.
This is what you're talking about?
yeah, there's these different readings, and it's a big finding. Perseverance on the rim of Jezero Crater
Yeah.
has been
So
picking up rocks and looking at things, and there are these tiny green toned, like they're these specks, right? And
okay,
so it is
the like
chemically
leopard spot
reduced.
looking things.
Yeah, there's chemically reduced iron in
Uh-huh.
there. And so they've been looking, the rover's been looking at it. They have the Mars orbiter looking at it. They've been checking this stuff out. And so we've got these interesting olivine, pyroxene, basaltic rocks, and they're checking all these things out. They found iron sulfite, iron phosphate nodules in these rocks. And that's interesting because phosphate, very, very active part of biology, ATP, right? That P is phosphate. And so there are these clays with lots of stuff that is, we can attribute a lot of it here on Earth to biological activity, but we are not there and we cannot do the tests there that we have done here. So we need to bring
So
rocks back, is the bottom
this is
line.
the scientific equivalent of, so you're telling me there's a chance. Right? Like,
Well,
that's basically what this is. It's saying, like, okay, we found something that indicates that it is not impossible. Yay!
no, it's not just that though. What it is we looked at every way this could have happened that isn't
Mhm.
life, and we've ruled it out, but we
Here
can't
on Earth.
definitively say it's like. And part of what it is that for this transformation, chemical transformation, to happen in these little spots that have rings around them as though they originated in a center point and were sort of expanding out, would require an intense heat or possibly an acid to be in contact, and they don't have any source or way that
Right.
could have happened in the Martian landscape to produce it.
But what about, like,
So-
millions of years ago?
So even over millions of years ago, for this to be on that surface, the only way we see it done on Earth is there are microbes that have the ability to do that chemical transformation of the iron. But for it to happen just on its own in dot form means an incredible amount of heat would have to be just right there. Oh, and just right there. Hey, we'll do one more right over here, and we'll cluster them up. I mean, like, yeah, some Martian visitor playing with their new laser gun, right? Taking a couple of test shots on Mars before taking off. Sure, that's another possibility. But
Or
the exciting thing
long
is really that this study wasn't trying to prove life. This study was trying to
No.
prove all the other ways that it could have happened, and they failed to find one. So it's a little bit more than I
That
would
does
say
not
that-
mean that there is not one that we don't know about. And this is not talking about now life. This is still talking about, this is ancient. This is not now.
Right.
This is
This is
ancient.
ancient.
And
right.
so
Extinct
could
life.
have been extinct life, which we're still talking about. Could have been, but we don't know. And we
Right,
won't
but it's
know until
more-
we bring rocks back.
Okay, but I would say though, there's a chance scenario is more on the side of there's a chance it's not life. Is much more right now, is much more how this is weighted. And that's why there's all the excitement about it. Is that
I think it's exciting, but we're also wondering whether or not there could be life in the atmosphere of Venus, right? And that was a big exciting finding because of certain compounds in the atmosphere at a certain level. And since then, conversations have taken place that people have gone, oh wait, no, there are other possibilities that we had not considered previously. So So you're saying there might be a chance. Yes, Blair.
Oh no, there might be a chance that it's not life at this point. Might be a
Yeah,
chance.
no.
Life. Not life.
There is no finding of life at this time. To say that is like what you're saying, Justin, is not that's not scientific method
It is. Oh
hypothesis.
no,
No, it's
100%
not.
is. It's ruled out the alternate possibilities because it's disproving things, not proving things, and that's how science usually works: is you
It is
disprove
disproving.
the other
Mhm.
possibilities. It doesn't mean you ever prove a thing, but you disprove the other things, and that's what they've done, and that's why it's exciting.
They've
Anyway,
disproven a whole lot of things.
yeah.
So we still have so many questions about things here on our own planet that we have not, figured out all the answers to just by thinking we had all of our assumptions correct. But anyway, I'm just saying you can be excited. If you want to be excited about the possibility of ancient life having been on Mars,
Can
it could
can I
have been a
just, can I
possibility,
suggest,
but it
Kiki?
also might not have been.
Can I suggest you
And
might
we
be a
don't
little
have
bit,
evidence saying
you
yes
might be
yet.
a little bit jaded. Like right away, you
Me?
brought up past relationships. You're like, "Ugh, I one time dated Venus, and like, it just didn't pan out. Why would Mars be any different? I don't
Oh,
know.
that
Like,
one time I
planets
got
in this
excited
solar
about
system are all.
like life, arsenic life in Mono Lake. Come on, that
Oh
one time.
yeah, I remember that. I remember that. Yeah, that was a very exciting story.
Come
Yeah.
on, everybody. Be excited, but have a little critical thinking going on.
There is, and that's, but I wouldn't be getting excited about this one. I would be shooting it down with every possible way possible that you can. But this is a bit, they've eliminated so many of the things that would have eliminated it as life.
Yeah,
And
they
the
absolutely have.
heat and acid required to do the transformation and make it look like, what does it look like? It looks like a ringworm on these rocks. It's basically what it looks like. It looks like the form, visual form that you would expect from a sort of bacterial colony or of some sort of fungal colony or whatever Mars calls it, Martian life colony. So just the eye test, but then the chemical transformations are something that microbial life on Earth utilizes to grab energy by that shift. It's how life works on Earth, and it isn't
And it's
another.
our same solar system, and yeah,
Shouldn't be different. It should follow the same rules everywhere in the universe, really.
but we don't know all of the environmental factors involved. We can guess at a lot of things, but we don't know because we weren't there.
But it
And
will need it
we've
would
got our
need
robots.
it would need an unknown at this point to take it off the table, and there is that unknown. For all they know, we were pretty good at knowing. It would have to be a big unknown scenario that pulls this off of its life shelf.
Okay. Hi, Blair. You like this debate?
I mean, I have two conflicting kind of philosophies on this. The first is obviously there's life elsewhere. We're not that special, right? We have extremophiles on this planet. Why can't life live in other extreme areas? So, of course, there's life. But on the other side of it, I also have this, whole philosophy of we know what life looks like here, what the chemical processes look like here, what the artifacts of life are here. We don't know that all life throughout the universe requires metabolic processes like the ones that we have here. We don't know that the same chemical processes equal the same kind of artifacts that we have here. And so I think that's the part where these sorts of releases lose me, is there's a lot of assumptions being made that all of these different kind of elements of science work under the same laws that we are assuming are universal. And there's assumptions going on. And I think that is.
But to assume that life would be like, say, carbon based is also partly because it's like so abundant, right? When we're
Here.
looking at throughout the universe.
Oh, sure.
I mean,
I thought you meant carbon-based life is abundant here. Yes, okay, yeah. I see what you're saying.
I mean, the building blocks that we use for life aren't the like hard to find ones. We went with the most basic toolkit to get started. And so I think that's where a lot of the assumption comes from, is what would be the easiest path is also kind of the one that we took. We didn't take the hardest route to becoming life. We took kind of the low-hanging fruit at every opportunity. And so I think that's part of why that assumption. Like, I used to have that same, like, why does it need to be a carbon-based life form? I watched an episode of Star Trek once, and there was a talking silicon rock. Oh my goodness. but really, when it comes down to the building blocks and what you got to work with and how we did it was with the most basic, most abundant things that we could, that were present. And so it makes sense to assume that that's going to be the most replicated scenario, at least everywhere in the universe.
The researchers write in the summary of their paper. In summary, our analysis leads us to conclude that the Bright Angel Formation contains textures, chemical and mineral characteristics, and organic signatures that warrant consideration as, quote, potential biosignatures. That is, quote, a feature that is consistent with biological processes and that, when encountered, challenges the researcher to attribute it either to inanimate or to biological processes, compelling them to gather more data before reaching a conclusion as to the presence or absence of life. And then they say we need to get rocks and bring them here.
Bring the rocks
tests here. Bring the rocks.
That is what you would call a professional, credible scientist, basically saying, "We found life! Oh my god!" But they don't do wow them out emojis in their professional papers.
We need more evidence. Bring the rocks.
Of course. Okay. More scientists is like, I don't need to say anything else. I'm calling it. No scientist is going to do that. And we're supposed to get the rocks back.
Bring me Naledi.
We need the rocks back. So, speaking of Homo naledi, if you don't recall, this is a South African cave where a Facebook ad grabbed asked for brave, short, skinny archaeologists to go dedicate time spelunking in South Africa. A trove of bones was discovered in a very hard to reach chamber of the Rising Star Cave, and they came out with a very kind of controversial, I guess, paper. First of all, the finding itself is just sort of astronomically controversial because they've discovered a new species. They identified and named a new species. And this has happened before with a bone, finger bone, a jaw, partial skull, that sort of thing. They have thousands of bones, complete skeletons, the whole anatomical structure of this with no inferred missing bones anywhere. This is not how this field normally has to approach something. They find a partial piece, and then all of the research community analyzes this one bone or partial bone feature and tries to extrapolate. So they got a little controversial because they made some I guess the opposite of what you were just reading about potential we got to watch out for. It could be a bunch of things. They were like, oh, Homo naledi buried its dead. And we have proof because we found a cave full of their bones that are buried.
But there were a bunch of assumptions from that one as well. And it was a bunch of...
Well, that paper was a lot of conclusionary. If you read the paper, it is exciting, but there was a lot of, we conclude this, we conclude this, we conclude this. It wasn't what I would call the most professionally written paper, but it was also a paper that was announcing a discovery that had all of its work ahead of it. So instead of waiting 10 years to publish a paper about their find, which is often what happens in these fields, they said, we're going to publish everything we found, make a bunch of conclusionary statements, we'll figure it out later.
Cool. You all can
So
be part. You just come along for the ride, everybody.
oh, and they did it. They brought some money into through a Netflix special and did public talks and sold some books.
Yeah.
And people complained about that, but a couple of things
Well, that
going
was
on there.
questionable, but yeah.
Well, questionable if it was in the United States. So Lee Berger is running archaeology in South Africa, which doesn't have the most resources being thrown at science. Not that America can talk anymore, but a couple of things. First, he wanted to make sure that all of the local researchers from the university and from the region were involved and had stakeholder roles throughout all of this. Also, by bringing money in, they can to his organization, they can keep doing more research internally. So instead of just we're going to send these bones to the US and the UK or whatever and have you guys analyze them and write your papers on them, he was being selfish in a way, but also like supporting scientists and supporting the research. Anyway, now this time they've got I don't remember, like 6 different nations of researchers, 20-something, 30-something researchers are all authors on this new paper where they've gone and done a much more thorough deep dive. And the conclusion, the main conclusion about the burial, which is what this paper is focused on, stands. They have ruled out water flow. There does not seem to have been water flowing at any point throughout this cave. So it hasn't been like the bones were somehow deposited there and then just covered up by water carrying bones or water carrying dirt and sediment over them. Part of that is because a lot of the bones are fully articulated. So you have like the whole arm with the wrist right there. So these were bodies that were placed there. There's no signs of animal activity or anything like this. They haven't been disturbed. And then some of the bones, everything will be articulated. And then there's a bunch of missing stuff with another burial in that place that's been disturbed, which indicates that it was not only used as burial, but that there were multiple burials. So maybe even generational. It could have been there for, they could have been using this for hundreds, if not thousands of years, doing burials in this same cave, where they would go in and dig out and move some of the old bones aside as they placed the new ones in. Hominoleti itself, if we're unfamiliar, is kind of an interesting mix. It's almost the opposite of what you would expect. It's got it's about 5 feet tall, has hands and feet and kind of lower legs that are very much like a modern human, but its hips are more like an Australopithecus. Like it skips Homo erectus. It goes back to a more apish hip structure, shoulder structure. So it's one of these weird things where if you had taken if they had found just the hip, if they had found just the hands, you would have ended up extrapolating a very different species based on just the few subtle clues, right? That you would expect.
Yeah.
But no, because they've got complete skeletons, they can completely define Hominoleti.
They have so much too. It's just a massive database. Like the find is just, there's lots of bones.
And then part of this paper too is introducing the fact that they've opened up a couple more chambers within this cave system that also have burial bones. There's some allusion to a stone feature that looks like it was intentionally placed, but they say there will be more analysis on that in the future. And there was a time frame that I don't think was in the original that I think they've narrowed down for when. And I think now there's a minimum age on this of around 240,000 years. So let's see. Let me see if I've got that right. Yeah.
That's great.
So it's over now. They know it's over 240, possibly 300,000 years. So this would have meant that somewhere in Africa, very early human Homo sapiens, current modern humans were running around already, doing their thing. Would have been one of, I think, 8 hominins, human hominin type things wandering
At the
around
same
the planet.
time.
Yeah. Maybe
love it.
9, because there's like, I knew, there's other, like we keep finding other ones that are like, oh, there's like that skull in Greece. It's like, well, it doesn't quite fit anywhere. It's a lot of doesn't quite fit anywhere. It looks like there was a lot of humans that almost all went extinct. And the thing about this one is too that I find interesting is like if they've been using a spot for burial over and over and over for generations, where were they? They weren't living in this cave. There's no signs of them doing. There's like no food bones and none of that stuff. They weren't living in the cave. They were depositing the dead here. Where were they living? What were they doing? Somewhere on the surface.
So I'm not saying I have any hypotheses based on this, but I just kind of want to mention that elephant graveyards. Do you guys know anything about that? It's
Where all the bones
talked about
end up?
it's talked about in like stories and in myths and stuff where, elephants go there and they mourn their dead and all this kind of stuff. But really, the situation is that elephants have a finite number of teeth. They grind them down throughout their lives.
Mm-hmm.
And when they get old, pretty much the only thing they can eat is squishy swamp food. And so old elephants go and they eat the squishy swamp food and they eventually die. And so they all die in the same place. So I'm just throwing out there, like I said, I don't have a hypothesis. I don't have like, maybe it's this, but it could be that they are putting dead individuals into this space. Or it could be that infirm individuals are going into this space and then die.
So
right?
some of them are
or
children, some of them are adolescents, some of them are teenagers. There's a range of, and that's why they say it looks like a graveyard, because in a graveyard you tend to see, like a battlefield, you should see all kind of adults. But this is the range of ages. And what have you. So that's part
that
of
makes sense. Yeah, I don't know. I was just like, man, was there like a mushroom in here or something that they were like going in there looking for? Or, it just I'm just throwing out there
the fungus. Yeah.
So this is the earliest. This is the earliest now, I think, burial, much earlier than current modern humans. But Neanderthals, there were debates for a long time about whether burials that we were seeing with Neanderthals were intentional or not, and it turns out we found now more and more examples that Neanderthals did occasionally bury their dead, and I think it was Lee Berger at some point is the one that was putting this forward, but it's not necessarily a supernatural connection. It's not necessarily even a sentimental completely connection.
It's hygienic. Yeah,
it could be a combination of hygienic. You don't want to attract predators. Right. And maybe the first time that you see a dear loved one who was, left out on the open tundra there, or what do you call it, in the plains there?
The savannah.
And they got the savannah. Thank you. They got torn to pieces by a pack of hyenas wandering off with their head and their arm and their leg and whatever. It was so traumatic that whatever hominids were like, from now on, let's do a covered thing where we don't have to see that again. It could just been, yeah.
And if you think about, this is earlier than Homo sapien or our earliest, the human burial, but we keep finding pieces of evidence pushing everything back related to our social activity. We know that there is concern about predators. We know there's in social groups, there is evidence for actions by social animals, by the upset, by like psychological, upset, mental strain in other primates that don't have language, that don't have the problem solving that to the level that we do. So if they are thought to have had a growing brain case and something that is very on the way to being totally human, or comparable. Why not?
Yeah. And then there was the
Yeah.
one last thing. The brain is kind of on the small side, the brain case,
Yeah.
but the internal foldings
Mm.
of those small brain cases look very much like a current modern human. And that's one of the other like really cool things, is they have so many like amazingly intact portions of the fossil that they can see the internal imprint in the skull. And they can see that the brain folding looked very much like a current modern human, even though it was a much smaller brain.
It was tidy.
Kind of like even like the Homo naledi, or not the Homo floresiensis is like, they're like 3 feet tall. Like I almost can't even picture, he's skinny, 3 foot tall with a tiny, even smaller brain. But they had evidence of tools and spears and things. So there's a lot of our preconceptions of what we would expect have been overturned in the last 20 years when it comes to what evolutionary points in human development would be required for us to do this, that, or the other. So
Yeah. Well, let's move on from human evolution and development to koala chlamydia. Blair, what you
Yeah,
got?
koala chlamydia is ready to be treated in the bush.
isn't that where it's always?
Yeah. It's the first vaccine for koala chlamydia is ready to go. This is developed by the University of the Sunshine Coast, and the approval by Australia's veterinary medicine regulator is the next step in this fight against what in many cases is the greatest threat to koalas. This has been over a 10-year project developing the vaccine to protect koalas. And in many cases, chlamydia can end, of course, in UTIs, but also in infertility, huge impact to the species, blindness, and it actually can kill koalas. And so this is now looking at a single dose vaccine with no need for a booster. So you could grab a koala, give them a quick stick, maybe give them a notch in an ear or something like that, so you'd know that they've gotten their vaccine and they are done for their life. That's it. And so you could potentially target areas where infection rates are high. You could target individuals that are approaching breeding age, because it is a sexually transmitted disease. And so there are ways that you could kind of strategically plan on how to administer these. Obviously, administering individuals with vaccines in a wild population is not ideal. Like, you want to figure out an easier way to do this, but koalas, their populations are just plummeting and have been for the entire time that I've been on this show. So it's been, almost 2 decades that these guys have been really struggling. And so this 10-year study is really showing some great promise to save the species, honestly. In some particular colonies, infection rates are as high as 70%. In a lot of cases, these vaccines have been really, really effective. They started on captive animals and in animals that were from the wild that were being rehabilitated. And now they are looking at deploying this in the wild. And so in their study, they found that it reduced the likelihood of koalas developing symptoms of chlamydia during breeding age and decreased mortality, this is crazy, in the wild by at least 65%.
Whoa. That's huge.
Yeah, exactly.
Wow.
And so they had hundreds of test subjects. It looks ready to go. I think the thing that is important to mention is, as is the case with a lot of human STIs, the main plan of attack is antibiotics. And particularly with koalas, antibiotics are a huge issue. because, as I am sure we've talked about many times because I find it so fascinating, they have a very particular microbiome to be able to eat eucalyptus. If we tried to survive off of eucalyptus, we could not. We would die. And it's because they have a very particular microbiome that allows them to, A, not be poisoned by it, but also B, actually extract nutrients from this plant. And so if you give them a systemic dose of antibiotics, it wipes out the microbiome in their stomach. And in some cases, the individuals can fully starve and die after having their antibiotics. So you've cured them of the chlamydia, and now they die because they can't eat eucalyptus anymore. So
tragic.
A vaccine is a much better choice in this case, but also because as an STI has a very particular way of spreading. So if you can apply a vaccine prior to them reaching breeding age, then that means you're not going to have the same spread each time they copulate throughout their life, right? If they're infected. So that's reducing the opportunity to become a vector of this STI. So, this is huge. And of course, Terry Irwin from the Australia Zoo Wildlife Hospital, yes, Steve's wife, she is part of the effort to get the word out, to fundraise for this, and to actually develop an implementation plan in the wild. And so I am very excited to see what comes of all of this, but I love the idea that we might actually be able to see some positive movement on the koala chlamydia front, because as silly as it is to talk about koala sexually transmitted diseases, in this case, it really was a threat to their species. So, yeah.
I wonder, I mean, we've got the, what's the animal in New Zealand that has the facial tumors that...
the Tasmanian tiger.
The Tasmanian the Tasmanian devil.
Or sorry,
right?
Tasmanian devil. Yes.
Yeah.
Not the Tasmanian tiger. That's extinct. Sorry.
Yeah, so that also is like a hepatitis, or like a transmitted by contact.
Yes, it's transmitted via bite, but yes, absolutely.
And so finding these endangered or threatened animals, that there is something where we could possibly relatively easily, now that we've found this, be able to treat and combat things that kill them and reduce the populations even more and make them healthier and happier. I think it's great.
Yeah, and I don't know what the treatment options are, but when you're trying to vaccinate wild animals, if it's a single dose with no booster, you could potentially administer things via DART too. Like, that would be super cool. I don't know how you would mark them as having received it. That's kind of where you run into trouble.
The DART just stays in. No.
Right. I know, right?
Yeah.
It also gives them a little tattoo at the same time. I don't know. But yeah, it's very exciting.
Yeah.
Yeah, this is really a situation where the issue was identified quickly, I would say. Like, all of a sudden, it was a big deal. Oh my God, all the koalas are getting chlamydia. And there was a lot of fundraising.
Yep.
There was a lot of awareness. And there was a lot of research that started. This particular piece of research was 10 years of research that continued to be funded. It continued to be supported. And in the end, it resulted in a real solution. And so I think, we don't often get to have that part of the conversation. We get to talk about issues. We get to talk about ideas. We get to talk about trials. We don't get to talk about, hey, we might have actually found a solution to this problem, and we might have had enough study done that we can actually implement the solution now. And so I do think that's really exciting. And so I wanted to bring everybody a little bit of good news on that front.
Thank you for the good news. We all need good news.
So at least in Australia, koalas can get their vaccines.
Thanks for that, Jab.
Anyway.
Moving on. Let's talk about some new directions we're looking at for treating cancer. So researchers have been looking at the microbes in the gut for a long time, and they have identified microbes that hang out with tumors in the colon. So when you have colon cancer, tumors in the colon often have certain types of bacteria that tend to hang out with those tumors as well. Some of them slow down tumor growth, and others speed it up, right? So it's always a question of what bacteria are there, what are they creating chemically, like how are they signaling and communicating with the tumors to be able to get what they need, because it's really all about the microbes. What they have discovered with this this study that was just published in, I think, Cell Systems, or what is it? Yes, Cell Systems. It's a Cell Press Journal article. This particular study, they were able to do a high throughput 4-way screen to be able to discover a particular compound that E. coli associated within C. elegans. This is not humans, but they did this massive C. elegans throughput 4-way screen. They determined that there is a molecule that is released by the E. coli that slows down the tumor growth. And in slowing down the tumor growth, they were like, hey, this is really interesting. And so then they were like, what does it do and what is it like? And they determined that this molecule also works along with a common chemotherapy drug that is called 5-FU. And I think that they acronymed it as FU. I know there is scientific naming, but I think that FU is just that, saying that to cancer. That is my guess. Anyway, the researchers were able to, through the throughput, be able to determine that these two molecules, the one created by the bacteria and the drug that is already in use, they work together synergistically to actually make anti-cancer better. So together they get rid of cancer than either one alone. And so they then were like, hey, I wonder if we can take this natural E. coli produced compound and make it synthetically. And so they were able to make it synthetically, and they found that their synthetic version actually worked better than the E. coli version. And so that testing it in all sorts of systems, they were able to show that this compound made by E. coli and this chemotherapy drug 5-FU together are able to reduce cancer tumor growth better than either one alone. So maybe we will be getting more of our drugs to fight cancer and other diseases from our microbes. We just have to look at what they're doing.
Yeah. Well, I mean, our old approach was just pretty barbaric. We will kill all of the weak cells.
Yeah.
We will cut out the affected areas, and then we'll wait for a few centuries until something else comes along.
Yeah. It was like quite literally the nuclear option, right? That's what we were using before. When isn't excellent medical treatments just waiting for us in nature? Here we go.
Put them together. Find it in nature. Now, sometimes we just come across things on our in nature.
Yeah.
Usually. Yeah. Anyway, there's potential to develop new drugs from natural microbial products. What you got, Justin?
Okay. Has anybody ever noticed that bees are social creatures?
Yes.
What?
Maybe.
Well, it turns out it's true.
Yeah.
Bees are social creatures, and apparently I no longer have my story. Hang on one second while I have technical.
I have it.
I know. How do you have it? Because
I have
to go reopen it.
I clicked the link. It's in you put it in the rundown.
Put it in the rundown, but then I closed it somewhere. Okay. So this is basically a genetic analysis of bees, and they found that a lot of the genes in bees associated with their social individual variation in sociability seem like similar genes that we would find in humans. Transcriptome sequencing revealed over 900 genes that were more highly expressed in the brain, or the more frequently a bee interacted with the nest mates. Whole gene sequencing of 357 bees revealed 18 genetic variants associated with the tendency to share nutritious liquid with nest mates. Yeah. And they share similar sequenced genes that have previously been linked to autism in humans with some of the variants that they found. Are bees just autistic? Is that what this is?
I don't think so. I think these genes they're probably involved in this social behavior, and there may be more expression of them or different expression of them
So,
in individuals who are autistic, or maybe it's how they're expressed in the human brain.
so what is really wild to me about this is if there's a genetic underpinning for sociability that bees have
Mhm.
and that humans have, and they have some sort of structural overlap, or they seem to be similar genes doing similar things.
Mhm.
Part of it is like, well, these are instruction proteins maybe that are being sent out, and the basic machinery of brains is not that far removed. Maybe it's something like that. But it could also mean you could also maybe extrapolate that there's a shared origin of sociability, that you would have to go all the way back to the common human and bee ancestor.
It's kind of way back there.
Exactly. So I need to see prairie dog brains and their genes and what's happening with them. I need to see
is it convergent evolution, or is this
naked mole rats? What's going on with them? Right? So pick kind of these like social networking animals. Ants. What are ants doing? And see where the commonalities are, because otherwise, is this just convergent?
Well, it's been 600 million years or so ish since we were related to bees. Like since our bee and human ancestor were the same thing. And then you're talking about possibly the basic infrastructure of all social insects, social mammals, social everything on land, having the same sort of set of instructions about working together. Now, that doesn't seem like it would be too far-fetched. My gosh, microbes seem to have levels of interaction, but this is at the brain level, right? This is something that has been preserved and is part of what brains, as the controller of the individual life form, is working with a set of instructions that go beyond the individual. Right.
And bees, calling what bees have a brain is kind of tough too. It's like a ball of nerves. It's like not quite. It's
the ganglion.
Brain is like very generous. Talking about what
but they do so much with it, right?
Yeah. Oh, absolutely. I'm not saying that it's like not as an insult. I'm just saying structurally, it doesn't really look like a brain. Yeah.
But this is chemical, right? These are the molecules or the genes that are in there that set up the molecules, the proteins, the enzymes, etc., to trigger stuff, to make stuff happen. And we have seen that there is a lot of conservation between different species over hundreds of thousands of years, that there are basic needs for connecting with other individuals for survival, and with bees especially, this is involved in how they find food, right? So there's going to be, if there was basic coding for chemicals that were involved in some kind of a pathway that led to positive reinforcement for connect, like sharing information in some way. I'm not saying I know how that would work necessarily, but yeah, but it makes sense that there would be conservation of certain instructions. But that's the question. Is it conservation?
Right,
Or
because
is it-
convergently, you can sort of picture those triggers, those mechanisms when you create a protein, when you have a gene that's creating a protein that's going off and eliciting work. That if it affects things in the right way, then maybe that is a convergent thing. Maybe that gene just pops up a lot of places, and because it's successful, or it's shared, or life has always been social. And actually, the real anomaly is the unsocial creatures. The unsocial forms of life are really the ones we got to start looking at, going like, well, why doesn't that life form like other life forms of its kind? Why is it such an antisocial life form?
Everything about life is seeking, is gathering information from the environment and then preservation of the organism. And so what are the things that need to happen to preserve the organism and to send out the genes again, right? To transfer, to reproduce. And so all those things are the basics, and that is going to be, we found it's, if you're alone, you're probably going to die because you're not going to reproduce. So you have to, at the basic level, begin, well, you're going to die eventually, unless you're like a sea urchin or something like that. But you're going,
or a clam. What is it? The oldest life form is that like 500-year-old clam they found in like Norway or something.
Well, and jellyfish are immortal depending on how you look at it.
But if the goal is reproduction, survival and reproduction, you're eventually going to have to figure out a way to be with others like you. And you're either going to be with them because you all spawned in the same place and didn't then go other places, or you're going to have to find them, or you've all come together in a way to live together communally, socially, right? So there's like these different solutions to the problem. But yeah, I don't know. So the solutions, there's base code, and those codes, there may just be a limited set of instructions that have evolved and adapted and mutated, to get where we are today. But is it conserved or convergent? That's what I want to know. We don't know. The answer isn't there, is it?
Oh, and can we create a drug that makes people more social? Is it a
because we have a loneliness epidemic?
Isn't that just alcohol?
It might already be a thing. Might already be.
Oh no. Oh no. Hey, just a quick question, because we're in the Pacific. I'm in the Pacific Northwest. You're a little bit further south of me right now, my friends. But do you like earthquakes?
Love them. Love them.
And you ever think about earthquakes and how much energy an earthquake actually has, and how much energy is going to the shaking of the earth versus other things?
Interesting. I mean, I assume quite a bit, though it is just all on like gelatinous soup. So
right, the surface is like where the ground shakes, right?
I don't know.
What is the thing? If the planet was an eggshell, or the planet Earth's mantle compared to an eggshell, the eggshell would be like 4 or 5 times thicker than the Earth's crust is actually on the planet. Like that's what a narrow band of crustiness that we are living upon.
But that crustiness, we have plates of crust, so separate pieces of crust that have been put on top of the pie or the jello or the egg or whatever.
Yeah. Or they go like this.
They come together and they smush into, like Blair's doing smushing up, or they're smushing
or sometimes
down.
they go across each other.
They slide. Slip faults. There's subduction.
Transverse fault is this one, I think.
Exactly. So at MIT, researchers have been creating lab quakes, because you can't really go and measure all the factors related to energy in an earthquake as it's happening in the crust of the earth. That'd be hard. So they took a bunch of granite and they powdered it up and they stuck it into a pressure thing where plates would like smush these little powdered granite bits together, really like a I guess like two plates coming together to see what would happen. And they had piezoelectric detectors to get motion. They had magnetic particles in the mix as well, so that they could use magnetic changes, polarity changes, and shifts in the magnetism to infer heat and energy transfer in different ways. And the researchers eventually have determined by creating earthquakes in the lab that what goes to the shaking is really not much of the energy at all. Maybe about 10% or so. You can get about, I don't know, maybe 20%, maybe 30% max to the shaking. The rest of it goes to the slipping and the movement, and then the heat. And the majority of the energy in an earthquake actually leads to heating the area around the epicenter of the quake. And when they were doing these experiments in the laboratory, apparently they were able to create slippage and movement within their lab quake that heated up so much that it was hot enough to melt material and create glass. So the stuff that they saw under the microscope was very indicative of the kind of rocks that we find in areas where earthquakes have taken place once we go and look. And so the fracturing of the rock, the way that that crystals and shards are formed and cracked, and what happens to the rocks. Anyway, they're now creating lab quakes, and it's not, a regular earthquake, but the way that they're doing it, they're hoping that they can understand what's happening in earthquakes a lot more to be able to figure out if one area has had an earthquake before, what does that mean for future earthquakes, depending on how the area heated up, how the earth in the crust may have changed as a result of that heating, and how it may have impacted a phase change or some aspect of the rocks. Anyway, they want to extrapolate, but right now they're doing lab quakes. Lab quakes! I took myself away. Why'd I do that? I don't know. Hey, everybody, this is This Week in Science, and we are just here for a moment to say thank you so much for listening to the show, for being here, and for appreciating the science that we bring to you during the show. We love talking about science, and we love talking about it with you. Thank you for supporting the show and being part of what we do every week. Our science is supported by listeners like you. So if you are hoping to help the show, please head over to TWIS.org, join our Patreon community as a supporter. And if that is not something that you can do, please share the show with someone you know. Thank you for your support. We really can't do it without you. All righty. Coming on back for that amazing part of the show that everyone looks forward to week in and week out.
Is it the end already?
No, jeez. Yes, no. It is Blair's Animal Corner.
Oh, yeah, with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I want to tell you about ghost sharks.
Oh, yeah, ghost story time.
Yes, they are a deep sea fish. They're in a group known as chimeras, and they're related to sharks and rays. They're cartilaginous fish. And they have a weird rod that juts out of their foreheads, the males do. And they have what appear to be teeth on top of them. So just picture, if you will, a weird deep sea shark-looking fish with a rod sticking out of their forehead that has teeth on the end.
That
So,
sounds a little bit odd.
A
yeah.
nightmare unicorn.
Yeah, I mean, it's not unexpected in deep sea fish to find just very, very strange looking things. This forehead rod is called a tenaculum, which is also, I was looking it up, that's also the name of a medical instrument. And so the word tenaculum means is from Latin tenere, to hold. Tenaculum in the medical field is a holding instrument. It's basically just like it's not quite forceps, but they look kind of like forceps. It's a surgical clamp with hooks at the end. So you can understand why this is called a tenaculum. But anyway, this all comes from the question, why the heck do they have teeth on there? Are they actually teeth? Like, in terms of development of this animal from the fetal state, like, what's happening here? Genetically, what's happening? How do you have teeth on there? Now, the first thing that I thought of was, well, sharks are covered in dermal denticles, which means skin teeth. And so, is this just more of that? Because that seems like a very easy jump. Sharks have skin teeth. This thing has teeth on the end of a bony protrusion on their forehead. Sure, right? And so that would be kind of like the path of least resistance for understanding what's happening here. But University of Florida researchers looked at this thing. They included some scientists from the University of Washington, University of Chicago. They studied fossils and living specimens. They looked at a 315 million year old fossil that had a tenaculum attached to their upper jaw, bearing teeth. And this was similar to those in their mouth. So it wasn't on their forehead, it was near their jaw. This appendage is most likely used to hold the female in place while mating, which males can do this with just their mouths in other shark species. But in this case, it's like a hook for keeping the females there during mating. But so then they also collected current specimens from Puget Sound. They looked at this thing on their forehead. And so then they also looked at genetics. And they found that the teeth do look like rows of shark teeth. They don't look as much like the skin teeth. They were able to make teeth that were similar to the teeth from the mouth. So when they looked at kind of like the development, it looked the same as mouth teeth development. And that they looked at CT scans of the fossils and the modern individuals. They really just looked like teeth, not skin teeth. And then they looked at genetics, and they found that the tenaculum teeth were using the same genes from true teeth. They had nothing to do with the genetics related to dermal denticles. So these are just teeth. And so this is
They have a toothy appendage.
yeah, and so this is an example of what they call evolutionary tinkering, or bricolage, which is like just like,
Isn't that an artistic technique?
yes, it's just like, it's just evolution being like, what about this? And so this is the super weird application of genes that are used for something else entirely. There's like, what if I put teeth here that worked for this group of deep sea individuals? And so they kept them. And so there you go. There's ghost sharks with a tenaculum with teeth
Tried it out.
coming out of their forehead. Yeah.
Tried it out. Seemed to work. Kind of liked it.
Mhm.
I survived and thrived. Let's keep it.
Yeah. And then
Curious
moving
to know,
on.
how does it bite?
Yeah.
Or does it just like stab? Like,
It's I assume it's a flick situation. Yeah. It's like a stabby flicky of just like, whoop, gotcha.
yeah.
Yeah, they don't have, it doesn't move. It's just, it's a unicorn horn. It's just sticking out of their head. Yeah. Yeah. But yeah, I didn't realize I had kind of a theme to my animal corner today. I have strange evolutionary adaptations, and why the heck does this animal have that? So this, yes, I love the ghost fish and their teeth coming out of their forehead. I want to talk about the matador bug, Beta ellipes.
Okay.
They're from Panama. They have red flags on their hind legs, and they perform an intricate leg waving display. And so if you were going to guess why might this animal have these red flags on their legs and wave them around in a special fashion?
Over here. Hey.
Yes.
Hey, over here. Hey.
Justin.
Hey, you, over here.
Come eat me. Yeah.
Is it come eat me, or is it,
No.
what do you think it is?
Hey, ladies.
Well, my guess is they're going to be living somewhere where visibility is not great. And so they're going to be like, hey, I'm over here. Hey, where are you? Oh, I'm over here. Because they probably don't know how to talk.
So the assumption would be 9 times out of 10, if you see a colorful display that is partnered with some sort of like dance or action, that is going to be to get the attention of females. So that is the expectation here. And that would be the kind of open and close shut case normally of just like, that's what this is. But hey, why am I bringing you this story? It's not that.
No, actually, wait.
Yeah.
So we just showed a little clip of this thing walking, and it wasn't what I was picturing. What that looks like is almost like berries or something.
So, interesting. So it's the jury's still out on exactly what's happening here, but let me tell you what researchers have figured out. So, first of all, kind of falsification of this idea that it has to do with a sexual display. Both males and females have them. Not totally unheard of when, it's just kind of like a sexy son hypothesis where it just so happens the genetic shakeout where the females have it too. That's possible, right? But that's kind of your first clue that you have something else going on here. Both males and females have it.
I want to rephrase because now we played another clip and I got a better look at it.
Yes.
In that clip. And it looks like a reddened leaf. Like a leaf that's like lost its green, like a decaying leaf.
Mm-hmm. Yeah, but it's a vibrant color. It's a weird shape to have in their legs. It's very unusual. And so if it's not for sexual display, why would they have it? And what Kiki's showing us right now is a video of this matador bug waving his legs in a very particular way at a praying mantis. And as we know, mantises are famous for eating everybody. Yeah. And so the flags appear to have something to do with deterring predators, which let's have a reminder of a couple things. So the first reaction to that is like, well, why would you want to call attention to yourself in front of predators?
You want to hide.
In most cases in the animal kingdom and in real life, if you see a threat, you don't want to be seen. So you'd want to kind of blend into the background. But as we know, there are brightly colored animals that display warning coloration for predators. They can say, don't eat me, I'm poisonous. Or they can look like the eyes of an owl. I'm bigger than you are, actually. Stay away from me, right? So there's all sorts of reasons. There's mimicry. There's all sorts of like warning patterns they can have. So there's all different signals that animals can provide to predators that just says like, I'm not worth it. Leave me alone. And so researchers looked at these matador bugs and their reaction to praying mantises, predators, and katydids. Harmless, just conspecifics, just like, hey, we're on the same leaf. Sup. Right? And so they looked at 3,000 leg waves. On average, bugs increase their waving behavior 7-fold in the presence of mantids, barely ever responded to katydids, and mantids never attacked bugs while they were waving. So this is a signal that mantids are receiving and going, gotcha, have a good day. It's an anti-predator behavior. But the question is, why?
How's this work?
So, yeah, exactly. So, these flag-waving insects, they eat passion flower vines. They carry toxins. They could be advertising that they are poisonous, like I mentioned before. We don't know that for sure. We don't know if the passion flower vines are specifically poisonous to mantids. We don't know kind of what the indication would be that where they'd have to wave their legs instead of just looking a particular color, right? Like, what's the addition here beyond just looking like they're poisonous?
So then the guess, if I can jump in and
Yeah.
guess, would be it's doing something particular to the mantis vision that just being a stationary color alone wouldn't have achieved.
Great. So the second hypothesis they have is that it actually is confusing their vision somehow. So it has nothing to do with the toxicity, but the fact that they have these flags on their legs, they are a bright color, and they are waving them could, in fact, be kind of giving them the dizzies and going, I gotta go. So that is another option. Is it intimidating them? Are they mimicking a predator? Is there an exaggerated motion that's making them look bigger than they are, making them look like something else entirely? There's lots of opportunities here that, they could be taking advantage of. For whatever reason, some of these bugs had fat legs that were bright colored in some mutations, and it worked. And so, it's the combination of the shape, the color, and the dance that I think is really interesting, because it is a complicated display that usually we only ever see in sexual selection. We don't see this kind of performative display with anti-predator action as often, I would say.
And to that evolutionary point, it might have been how it started. Right? It may have been a mate display, but it turned out like its most effective use was preventing predation.
Right. Right. And so when females occasionally had them, then they survived. And so they carried on the genes of females having them. And so, yes, you could totally see how that would kind of snowball over time, that it was like a happy accident that they did it for sexual selection. And then it ended up being helpful in preventing predation. There's lots of pathways. We have a long evolutionary runway. Yeah. So it's, it's been a long time.
To know everything about mantis perception.
Right.
Like, do they have like a persistent or slow processing speed, I guess, on vision, so that this waving that we can see clearly is like moving a thing becomes a single large blurred object, perhaps. Right? Like now we need to see things from the eyes of the mantis to see how it is affecting their decision making.
Yep. Yeah. These are all great questions, and I can't wait to find out more.
Oh, wait, I thought you had the answer. I was trying to throw as many speculations out there ahead of getting the actual answer. But I have to wait? Oh, no.
They've got to have, I'm just thinking, they have to have great vision. I think praying mantises are known for having really good vision because they're predators, and they're very often not striking out at stationary prey. They are stalking prey. They're going after prey that's moving. So I think that their eyes are really special in the insect world. And I feel like
it says they have 3D vision, a wide field of view, and excellent depth perception for catching prey. Yeah, you're dead on.
So they're going to be it's not going to be blurry. They're going to be perceiving the movement, but what is it stimulating or not stimulating? Is it
in a study that I am assuming I covered on the show? If I did not, it should be illegal. They made tiny 3D glasses for praying mantises back in 2018 to check their depth perception. This does sound vaguely familiar. I feel like I must have reported on this, right? I must have.
I think you
did.
That's
amazing. Yeah. That's amazing.
So
look at it.
So okay, so they have good 3D vision, but maybe maybe it's not like what we would consider the 3D vision. Because in that image that you showed, it wasn't just waving, but it was sort of moving things nearer and further away. Each leg was sort of moving these flags closer and then further away, closer and then further away. So maybe it was disrupting the processing of the depth perception.
Yeah, apparently, according to previous research, praying mantises have terrible red and yellow vision.
Hmm.
Is what
So
Well, those are the
colors
colors,
were.
right? Yeah.
Interesting.
So they have great.
Yeah, it's interesting they didn't
ultraviolet,
bring that up in the
green.
article at all. Yeah.
No, we'll get to the bottom of this before the end of the show. Keep
Okay.
digging, Kiki.
6 hours later, huh?
I know.
We'll figure it out. The three of us. Where's our grant?
Anyway, mantises, great vision, except not in color. So what are these yellow and red waving matadors doing? I love it.
Yeah.
Justin, what do you have?
Oh, I don't know. Do I still have more stories? Ah, yes. This is out of University of California, Berkeley. First ever measurements of ethanol content in fruits available to chimpanzees and their native African habitat shows that animals could be easily consuming the equivalent of more than 2 alcoholic drinks a day, according to the researchers at University of California, Berkeley. It's not clear, according to this press release, whether they are actively seeking out fruit with high ethanol levels, because these are also typically riper fruit that also have more sugars to ferment in them. So it may be a sweetness that's the allure. But the availability of ethanol, the fermentation process happening within these fruits naturally, that they're normally eating, suggests that alcohol is a regular part of chimp diet.
A regular part. Amazing.
Regular part. So,
Well, it's
that's
got your
kind of
carbs, it's got your sugars, it's everything you need, right?
And the buzz. And the...
Yeah. Just eat a
This
few
is,
bugs for protein. You've got a balanced meal.
Do chimpanzees have the same ability to break down alcohol, though, and the metabolic byproducts of alcohol? That's the question.
says if chimps are eating 5 to 10% of their body weight a day in ripe fruit, even at low concentration yield, high daily total, a substantial dosage of alcohol would be expected. So based on average consumption, they did some math. And they said, yeah, would it be the equivalent of a human having about 2 alcohol beverages a day?
So do they build up like a tolerance like humans do, right? So like there are some people who 2 beers are nothing, right? Like they could walk a high wire after that. They'd be fine. So it's just if they're doing this every day, then over time is it affecting them less and less, right?
Possibly.
Yeah, and
Or are they sleeping? They sleep a little bit later some mornings than others, depending on how
Also,
many.
how's their liver?
That's
Well,
what
because
I
you're
was going to
talking about
ask.
an average. You're talking about an average. They could very easily then, if you're talking about the average is 2 alcoholic drinks a day, you could have instances where they found a tree where a bunch of the fruit was fermented, and maybe they got up to like 5, 6 drinks that day. And then you have to start looking at how much of chimp society is based around alcoholic binge eating.
Yeah, like it's are
gonna be
chimps
during certain
more
seasons, right? Yeah.
more violent than other primates, or are they just hungover,
Oh my gosh.
lashing out?
More likely to be mean
Yeah.
after
Or
a night
friendly.
of
Maybe maybe they're friendlier. I don't know. You gotta figure out what kind of a drunk each chimp individually probably gets acts a little bit differently.
So I have a question I'm guessing you don't have an answer to, but I just want to throw it out there.
I'll have an answer. I won't commit to whether or not it's correct.
So these fermented fruits, the chimps have evolved alongside these fruits in these natural spaces for a very, very, very long time. So I'm wondering, like, when did humans invent alcohol, right? Like, pretty early in our timeline, but our timeline is much shorter than chimp timelines, right? So, like,
I'm sure, though, that we were making use of fermented fruits
right.
equally, but it's the, the manufacture or the making of alcohol, right? That's the difference. But
fair point. So yes, I guess we're not that different, but I was just kind of wondering, because if they have been living alongside these fermented fruits for, a very, very long time,
forever.
then how much is it actually affecting them?
That's a very good question. So this is the title of a book. The lead researcher, Dudley, wrote a book in 2014, The Drunk Monkey, or The Drunken Monkey. Why are we drinking?
They're not even monkeys.
This is, well, no, he's studying this in apes. But the drunken monkey hypothesis was the idea that chimps and other primates have been doing this for a very long time, and likely early human ancestors did, and that alcohol consumption in humans has an evolutionary basis, not just a modern add-on. That we've always been drinkers.
And was this study funded by the alcohol industry?
I don't know.
Don't stop going to your
but
local bar. I think it's just, I would like to know if the chimpanzees are honest with how many drinks per day with their doctors, like, when they go in.
No way. I mean, I'll have a fruit or two, just at the end of the week to unwind, just like normal stuff,
Just normal, just like anybody. Yeah.
Just to take the edge off.
All right, to take the edge off, I want to talk really quickly about the first insects known to clone another species.
You talking about plants?
Animals? Well, insects.
You said insects.
No, but the insects are cloning plants?
No, another species of ant.
Wow.
Yeah.
What?
This is a weird, wild story, and it's just fascinating. It published earlier this month in Nature. Researchers were looking at a species of ant called Mesor ibericus, and in the Mesor ibericus species, you have this is the Iberian harvester ant. All the colonies, the workers are hybrids. And the queens then have to mate with males, and these hybrids are sterile. The queens need to mate with males from a distant related species, Mesor structor, in order to keep the colony functioning. So all the males, all the hybrids, there's like none in her own colony that are going to work. And so she has to go to this other colony to get the males. Now, the interesting thing about the way their mating works is that there are not Messer structures nearby. These are like, they're someplace else. And so they were like, what is happening? I'm looking at these ant colonies. I'm looking at the individuals in the colonies. All we see are Ibericus and these hybrids, and there's obviously something happening here, and they're continuing on, even though the female, the queen, she shouldn't be able to mate, right? So then they started doing genetic testing of the colonies to determine what was going on in there, and they found that the queen, it only mates once in its life, stores the sperm from the meeting. It has a very special organ to do that. And those sperm are used to make 3 different kinds of ant eggs. You got other queens, workers, hybrids, or males. when they were looking at all of the genetics for all of the samples, 132 males from the Iberian harvester ant colonies, they found that there were different types of males. Half of them ish were covered in hair, others were hairless. When they looked at the nuclear genomes, all the hairy ones were Ibericus, the ones without hair were Structor. The queen had never been to find a Structor ant anywhere nearby. And so the queen is laying eggs for males of 2 different species. And so they looked and they're like, are there any Structor queens hiding in here? Where are the male Structors coming from? No, they're not. So they found that mitochondrial DNA being passed down from the queen. So they found that the mitochondrial DNA in the Structor males from this mating male that the queen would need from their mother, so Structor female, they found that maternal mitochondria in the mother of the Ibericus males. So they're like, what is happening here? They looked at all the queens, they looked at the eggs, they looked at all the stuff. 9% of the Ibericus queen eggs contained Structor males.
How?
Cloning. So the Ibericus queen has this sperm, and they've got the Structor males like in the colony, but the harvester queen ants are cloning the Structor males. They do not pass down their nuclear DNA.
Wow.
What on earth?
So this is like, what is a species? Like,
and how's she making an egg? Like, how?
I mean, I think, this is this whole concept of the passing on of genes and the creating of new organisms and the way we imagine everything coming together. But there is nuclear DNA, there's mitochondrial DNA, and these things are being passed down separately. And the fact that they're cloning Structor males, these queens are mating with one of the Structor male clones in the colony when they're in that phase for mating, storing that Structor male nuclear material and mitochondrial DNA that's a Structor male, not inserting her own Iberian nuclear DNA, and making more Structor clones.
So
hold on. So I'm a female ant. I make an egg. I take a male sperm. I take half and half through meiosis, right? Like I make 2 haploids or whatever, and then I smoosh them together, and I have an egg with a diploid set of cells, set of genes that develops into a genetic mix, right? If I am cloning myself, if I'm like a whiptail lizard, right? Mm-hmm. Then I am taking one of my eggs and just like I'm just splitting it in 2 basically with full sets of genes, and I am making a whole new one of those, right? So I will have it might not have the exact same genetic makeup that I have, because it might double some things that I only have one of, right, or whatever. But but I can do that with my stuff and make an egg with a gamete in it. Mm-hmm. What I'm not understanding is how I, as a female, make an egg with a gamete in it with only the DNA from a sperm.
Yep.
I don't understand how that works.
So this is
it a special process? Yeah.
Surrogacy.
They're taking, yeah.
She's a surrogate mother
for
the genomic makeup that is all found within the sperm.
Yes.
But how?
Clever.
It's amazing.
Clever. She's super, super clever.
I'm gonna make an egg and stick a sperm in it and shake it up. Like, I don't understand.
Kick out my own nuclei.
Right. Like, that's what I'm
this
wondering
one.
is
Yeah.
like, if it's like when it becomes time for combination, does the female genetic code get ejected? Like, how?
So the answer probably is in the structure colony, I would imagine. That there's something strange about its reproduction, maybe, that when there was this two different strategies that maybe somehow, and then when they did at some point must have gotten together, it had this anomalous effect. I mean,
And
it
it's
can't.
maintained. And they're like,
Yeah.
and okay, if you go and parasitize a structure colony and are able to do that, that's great. But if not, I've got my own clones around, and so I'm always taken care of. Make more clones.
Right, because that's the
Why
other
is it
thing is that
necessary?
it's not inbreeding per se,
No.
because the genetics are completely unique
Mm-hmm.
from the queen. But so it will create an over representation in the colony as a whole if you use the same genetic male over and over and over and over again to create your babies, right? So there will be kind of a replication of potential combinations, I guess, right? If you do that, everybody's a sibling. If you keep
Now I wanna-
mating with,
How
but she only
often do
needs to
they-
do it once,
Yeah,
right?
how often do they parasitize the structure colonies if they're not close by?
Or-
How does
or-
this happen?
or
When does
is-
this happen? Does it ever
Or
happen
is
anymore?
the structure colony an escaped clone colony? Right?
Oh God.
that went off and so they were only clones, and now they've gone off and started their own, like, free the clone, like escaped one day.
That is
Like there was a girl somehow at one point,
And that might have been that, could have been any species that they crossed with, but because all of the males have this, it just genetically overtook, and that became defined as structure, right?
Yeah.
That's wild.
Yeah.
Oh, I can't wait to find out more.
Yeah.
Wait, we don't have the answer again. Oh, it's another cliffhanger.
Yeah, but they are, they're
Yes,
this
Gord,
is
life,
a really weird
finds a
situation.
way.
Yeah. Life does find a way.
Yeah.
This is such a strange situation. Clonality, but not within the same it's mixing species as we define species, and as we like to categorize and split things up. They're not the same, and this is such an interesting situation.
Hmm.
But anyway, that's fun, right? Not so straightforward.
Man, thanks for bringing that, Kiki. What a cool
You're
story.
Reproduction is so wild in nature.
I know, it's all the people who want to make it so simple. Man meets woman, they have a baby. That's it. Nope. There is more scenarios than we even know.
I know.
Nature's complicated.
Scenarios you have never thought of before. Like I love the Live Science article on this, because the interview with the researcher is actually, he's like, we were joking about this. We're like, haha, wouldn't it be funny? But they didn't think it was true. And then they're like,
how do we publish this?
Oh, we need to test this. Oh my gosh, this might be true.
No. I bet you anything that's a very thorough paper, because it is one of those things like we can't publish until we're for sure.
Yeah. But as always,
goodness.
more study is necessary. And I'm going to take us out after that story with, a quick dip into scientific fraud. Back in 2003, one of the main scientific, what is it? It's a metrics for science leader, the database that Clarivate Inc., which is responsible for basically giving journals impact factors. And it's one of the groups that looks at how much have you published? How many authors are publishing? What's getting cited? And you're fancy. You're not fancy. They decide who's in and who's out of the science club and how many points you get. So Clarivate is not the only, but it's a very,
It's like a prominent one.
It's prominent. It's got like 10,000 employees. It's like making lots of money hand over foot, which again, that's also an issue. They're very not transparent about their methods, but somewhere back a ways, they took math, the entire field of math, out of their metrics. They stopped giving metrics any impact factors. They stopped doing the metrics for it. They had calculated their database. They did some work, and there's a couple of papers in the archive.org right now, preprints that go into a lot of detail on this. But Clarivate Inc. calculated from their database that in the year 2019, according to their database, the university with the most world-class researchers in mathematics is a Taiwanese university where math is not a subject. It's not a subject at the university, and it has the most world-class researchers in math. So anyway, hey, there's a problem in publishing and how we count what is good science and what's not good science. And the bottom line is that these papers in archive.org are basically reporting on the huge problem with parasitic journals, with fake publications, with people publishing work that is subpar and never ever read, aside from maybe a cursory peer review, that there is a lot of issue with the way that publishing is happening right now. And it's one of the things that they do is they've published actually practical advice, how to fight fraudulent publishing in the mathematical sciences. So they're focused on the mathematical sciences, but honestly, this is not just for math. Math is one of the smaller disciplines. It has a small number of researchers globally. It has a small number of publications. And so because of the small number, changes in statistics, like you can balloon your impact factor based on the way that things are measured very easily. Things can be put out of proportion really fast and really easily. So anyway, I'm gonna share this tab, but they go through a whole bunch of recommendations for policymakers. Why should you care? What can be done? For institutions, why should you care? What can be done? A lot of this comes down to, hey, if we keep allowing fraudulent publication and this misrepresentation of what researchers are actually publishing, it's not going to be good for the science that's being done. It's not going to be good for the science that's being funded and that funding. It's not going to be good for the public. It's not going to be good for your institution, because eventually that falsehood is going to come crumbling down. And additionally, for individuals, your integrity is at risk. Everything has an issue, but they have, what can we done for individuals? Read the actual publications when writing evaluations. Avoid publishing in predatory journals. Help educate younger researchers. Help identify good journals in your field. Maybe that'll help with how the impact factors are done. Cite articles that are relevant for your work. If a respected colleague is associated with a predatory journal, talk to them about it. Maybe say, hey, stop doing stuff with that predatory journal, colleague. Check the quality of journals before joining their editorial board. Choose the journals you publish in wisely. I mean, is any of this so far not common sense? But anyway.
Well, so
Don't be a fraud.
Don't be a fraud. But it's also beware.
Like,
watch out for
some of it-
phishing emails. Watch out for the misuse of names of things. Yeah. But anyway, go ahead. Sorry.
Some of it is like a little bit unfair though.
Yeah.
Like there's a whole beware and be careful where you publish and this sort of thing. Like the last few years of doing science writing, of getting studies from what is supposed to be the prominent sources journals and finding just ridiculous stuff. I mean,
what I do love though here, one of their strategic recommendations, changing the way that that publication happens and how you get into publishing, governments supporting efforts for greater transparency, culture change in academia. My favorite though, advocate and fund serious science journalism.
yeah.
That's my favorite. So I think they've come up with good recommendations. This is something that, is a challenge, and there are resources out there. And I can't wait. This is going to be published eventually this October, but for now, here it sits. Hey, science, you gotta mind yourself. Y'all have to step up.
There were two studies that I was considering for Justin Trash's a paper for this week. One was just an error, but I think it looks like it's just an error. It's another one of those. It crosses 0 and shows the highly significant p-value, and it can't be right. But if it's a negative value instead of a positive value, it's exactly where it's supposed to be. And so it could just be that somebody doing the data left off the negative sign at the beginning of one number, but it was repeated. But it was repeated multiple places in the paper, and that one's like probably not a big deal. Another one started by the fact that it was stated no conflict of interest, and it was a yoga method thing, but the organization that did this, that was involved in the study, two of the authors are specifically do studies for this one institute that also its founder is the one that created that yoga method. So it's a whole circle of like, obviously this is an in-house study finding a positive result. Plus it was an EEG, but they couldn't use some of the channels of the EEGs because they were getting too much noise. So they left them out.
Yoga.
Because they left them out. And so you have an EEG that's missing parts of the brain. And the way EEGs work is sort of like how it's a lot of bleed over into how the whole topography of a thing is working together. So if you've taken out certain things, you could have very different results. Yeah. There's all sorts of things, but some of it is like, that was sloppy methods that's hiding the fact that it's an in-house. The other one looks like an error. And there's the ones that, we talked about a couple weeks ago where it looked like they added a completely new thing to a study that obviously hadn't been there when the study was initially written, but they added something to make it have impact. And we're too sloppy to fix all the documentation. These documents are not a lot of pages. I don't understand.
Yeah. It's also
how you have this many
peer reviewers there's a lot of junk being reviewed, being submitted.
Can I just tell you, it's not being reviewed.
That's what I was gonna say. Peer reviewers, i.e. scientists, they're overburdened. They don't have enough time, they don't have enough attention, and there's too much coming through. So we're seeing, not a lot of like benefit to scientists to do the reviewing. So it's a systemic issue. We're trying to continue to report from sources that are peer reviewing, and we will tell you if it is preprint, if we're reporting on that. And we are going to try and give you the caveats as far as we know them, but we are trying to do our best to bring the best science research we can, unless we're going to tear it apart for you.
Unless we're going to tear it apart. But I think one of the impression I've gotten is that nobody's reading these studies before they get published. I don't believe that the peer reviewers are really going through them. I know the editors of a lot of these publications aren't going through them.
Either
And
they're not or they're sloppy.
the delay in finding a bad paper might be a few years from now when somebody looks up a paper that seems to be related to the research that they're doing, and then after looking through it realizes, oh, this is garbage. But at that point, the impact or whatever benefits you get from publishing to get your new job or whoever like did that, they've already gotten away with that. If you retract it, it's not affecting. So there's like very little downside to doing the fake publications, and that's part of the problem.
Right now, it's mostly upside for the wrong people. So yeah. Anyway, our system, so many things, but people are working to fix them. People are caring about this. That's why this paper is being published, why they've put it in archive as 2 papers actually as a preprint. But right now, I think we've come to the end of our show. Yeah, I think, have we done it?
I think
We've
so.
done it. Yeah.
Yeah. Yeah. Hopefully no one will have to retract this show later. No, never. Thank you all for joining us for another episode of This Week in Science. We're so glad that you have been with us for this tight 90, right? Anyway, thank you for being here. Everyone in the chat room, thank you for chatting, for enjoying, for being part of the conversation. For people listening or watching, yes, after the fact, thank you for being part of this experience and sharing your time with us. Fada, thank you for your help with social media and show notes. Gord, Arnlor, others who help to keep the chat rooms good places to be. I appreciate your help and thank you for being here. I think everybody here tries to keep our chat rooms nice. Rachel, thank you for not recording. Let's see. Identity 4, thank you for recording the show. And Rachel, thank you for editing the show. And of course, I can't miss an episode without thanking our Patreon sponsors. Yeah, thank you to Aaron Anathema, Adam Mishkan, Alan Viola, Ali Coffin, Andrew Swanson, Arthur Kepler, Artyom, Bob Calder, Bob Coles, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts. Craig Landon, Craig Potts, Dana Lewis, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Don Mundis, Jonathan Stiles, Don Stylo, E.O., Eden Mundell, Eric Knapp, Flying Out, Fredis 104, G. Burton Lattimore, George Corris, Greg Briggs, Howard Tan, Jack, Jason Olds, John Atwood, John McKee, John Ratnaswamy, Johnny Gridley, Ken Hayes, Kent Northcote, Kevin Parachan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessenflow Noodles, Patrick Pecoraro, Paul D. Disney, Paul Ronovich, Philip Shane, Pierre Velazar, Pete I.G., Richard Badge, Robert Norland, Robert W. Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean Clarence Lamb, Shoe Brew, Stephen Alberon, Steve Leisman, aka Zima, Sue Duster, Teresa Smith, Tony Steele, and Vegard Shefstad. Thank you all for helping out on Patreon. Thank you for being part of that Patreon community and supporting TWIS. If you would like to help support TWIS as a listener, as a supporter on Patreon, head over to twis.org and click on that Patreon link. On Friday, special episode, September 19th at 12:15 p.m. Pacific time, we have an extra episode for you all. We're going to be speaking with doctors Michael Mann and Peter Hotez about their new book, Science Under Siege. Peter Hotez has been prominent in the vaccine world. He's a public health professional and has been working. He worked on one of the COVID vaccines that has helped so many around the world. Michael Mann has been essential in a lot of our understanding and communicating about climate change. Michael Mann is the hockey stick scientist. So they've written a book together about their experiences and what is happening to science in the world. And I hope that you will join us this Friday, 12:15 p.m. Pacific time. But otherwise,
we'll be back again next Wednesday, 8 p.m. Pacific time, broadcasting live from our Twitch, YouTube, and Facebook channels.
Want to listen to us as a podcast, maybe while you're searching for life at the back of your fridge. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard today, show notes, links to stories are available on our website, www.twis.org. And you can also sign up for Blair's newsletter.
When
She
did this
tells you
become
all
mine?
kinds of fun animal facts.
That was recent, I think, while you were gone.
Great. Well, I'll
We had a
continue to
meeting.
deliver it in the cadence it was delivered previously. We love your feedback. If there is a topic you would like to cover or address or a suggestion for an interview, please let us know in one of our social media accounts, or better yet, send us an email. Just be sure to put TWIS, TWIS, in the subject line, or your email will be published in a bogus journal that no one will ever read.
We hope you enjoyed discussing science here today on This Week in Science. And if you learned anything from the show, remember,
It's all in your head. Nope. Did something happen to my...
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news.
And that is the end of our show. And I don't believe that we are going to have an after
Say goodnight,
show
Blair.
this evening. Blair is in recovery, and we all should sleep.
Good night, Blair. Say good night, Justin.
good night, Justin. Good
Good
night,
night, Kiki.
Kiki.
Thanks, everybody.
Good night, everyone. Thank you for joining us for another episode of TWIS. We'll see you Friday, hopefully, and if not, next Wednesday. Remember, stay sane. Stay safe, stay healthy, stay curious, and stay lucky.
I'm going to go find out why it looks like